We continue to run a little bit ahead of schedule, which makes me feel proud at this collective efficiency. Let's go on to the fourth of our scheduled speakers, Amy Pruden from Virginia Polytech, and will be talking to us about water systems.
Okay, since we're ahead of schedule, I'll try not to get too carried away thinking I have all the time in the world. But it is really an honor to be here and have the opportunity to talk a little bit about water microbiomes and a snapshot about why they're important and some of the work that we've been doing. But I do want to emphasize, as I start off here, how important a team effort is in research like this. So I've been working very closely with Mark Edwards, who brings expertise in water chemistry and corrosion and practical understanding of plumbing in homes, as well as Joe Falkenham, who's worked quite a bit in linking disease to people's plumbing, and Annie Pierce, who's a building scientist. And if it wasn't on the radar of normal everyday folks how important tap water is, I think it's fair to say that what happened in Flint, Michigan has really changed everything about that. And here is Mark Edwards testifying not once, but twice to Congress just in the recent months. And I just think it's gotten people's attention, the poisoning of an American city. And as far as the science is concerned, it's pretty basic that when you have corrosive water, it's going to leach stuff, toxic stuff from pipes. And that's experienced by consumers in terms of the water smelling and tasting and just looking bad. But I think there's also a lot more complexity here that we can appreciate. And just one thing is the fact that people's hair fell out and that they got rashes. And why is that? I don't know that we'll ever fully understand that. But the saddest day for me as a scientist was the day that our hypothesis came true. And that was that the switch to the Flint River water and all of the disaster that ensued in terms of the deterioration of the water quality. You had a lot of iron in the water, loss of chlorine residual, so corrosion eats chlorine, a lot of nutrients in the water, that would trigger proliferation of Legionella. And that was what was the key hypothesis of our NSF rapid proposal that we wrote last July. And we were astonished when we saw the news that, in fact, 87 people fell ill with Legionnaire's disease and 10 people died during the water switch. So this was one of those moments that normally when your hypothesis is validated, that there's some cause for celebration, but not in this case. And it's just crazy that this happened. And so what does this mean in terms of water and water microbiomes and water infrastructure? I think a lesson here is that Flint is not just Flint, but Flint is everywhere. And the American Society of Civil Engineers, as has been mentioned, has been harping for years that our water infrastructure is in really bad shape. And they've been giving it low grades. The most recent grade is a D. And I think people are really ready to take action. So I think that's one potentially positive thing here, is that people have realized the importance of water infrastructure. There's going to be action taken, and I think folks like us, the various scientific bodies that we represent, are going to have an important say in the action that's taken. And broader perspective, we have a lot of issues of concern with our water supply. So we've got population pressure and drought and pollution and climate change, all putting pressure on our water supply. And we've got some folks some bright scientists working on thinking about how would we reinvent our water infrastructure. And so an example is this NSF Engineering Research Center that's led by Dick Luthi and David Sedlak, and thinking about, okay, how would we ideally do our water infrastructure to address these many issues? And lots of things we do doesn't really make sense, like why do we flush our toilets with pristine drinking water? And as we're thinking about things like that, we have an opportunity to do things right. And importantly, as we strive for water sustainability, I think we have to be careful not to forget our primary mission as water engineers, and that is public health. And here's where the microbiome comes into play. So the primary cause of waterborne disease outbreak in this country are now opportunistic pathogens. So that includes things like Legionella pneumophila, Mycobacterium avium, and other organisms. These are very difficult to deal with because, one, they're atypical exposure routes, things like breathing aerosols in the shower. And also that they're not fecal associated. These are things that live and grow in plumbing, and that really makes our current framework for monitoring and regulating water quality pretty irrelevant here. And then another issue that's creeping on the radar of water quality is antibiotic resistance, and this is a big issue. Conservatively, we estimate that about 23,000 people die per year in the US of resistant infections, and about 2 million people are sickened. So a question is, could our water systems be a conduit for the spread of antibiotic resistance? And we made that case pretty meticulously in this recent feature article. But basically, you have people consuming antibiotics, excreting them, excreting resistant organisms. They end up in our water cycle. And what happens after that? So here's my first microbiome slide. So I don't know how many of you have ever had a chance to look inside the pipes in your home. Maybe plumber came over, removed some pipes, and you got to see what's inside. It can be pretty shocking. They're pretty slimy, and that is biofilm that grows on the interior of pipes. And there's a lot of microbiology going on in that biofilm. So just a bit highlighted here. So we have the opportunistic pathogens tend to reside there, and there's this life cycle with amoeba that will consume them and actually enhance their proliferation and their virulence. And also antibiotic resistance could be enhanced there. You could have gene transfer happening because of the close proximity, and maybe the chemistry of the water could stimulate the spread of antibiotic resistance genes. But over here I've listed several factors that can contribute to this. So things like disinfectant residual, your chlorine, the stagnation, the nutrients in the water, temperature, metals, other toxins. So these are all things that we might have control over, that we do have control over in a lot of cases, and maybe could move towards intentionally manipulating the microbiome. And definitely it's been a life-changing experience to be part of the Sloan community and working with the MOBI group. And they brought a lot of awareness of the importance of the microbiome, of the built environment, and how it's likely shaping the human microbiome. And I think that the water microbiome is relatively unexplored, but has profound implications. And so delving into one of our first major research questions here, we're really interested in the relationship between the nature of the water provided by a water utility and the quality of the water that's actually at the tap in your home. And if you think about it, a drop of water goes through quite a rigmarole as it leaves a drinking water treatment plant and then shows up at your tap. And a lot of that actually happens in the home where there's high surface area, there is stagnation, lots of complexity with pipe materials, and generally warm temperatures. And working with Mark Edwards, we have these standardized simulated plumbing rigs that we installed at 5 locations across the eastern US, and then we could basically separate the effect of the municipal water versus the configuration of the plumbing. And Flint is one example of why we're interested in that, right? There was this change in the municipal water, and suddenly the water at the tap was making people sick. And so our PhD student, Panji, did some elegant work here looking at the microbiome of these simulated premise plumbing systems. And what she saw was that the quality of the municipal water really was the overarching factor shaping the microbiome at the tap. But the premise plumbing was important too. So the pipe material, the stagnation time were like icing on the cake. And she applied a core microbiome approach and also did unweighted and weighted beta diversity analysis. And the take home there was that what was driving the differences in the microbiome in these different waters were the rare species. And pathogens are usually among the rare species, so that's not to say the rare species are not important. To delve a little deeper, we've also done work on the hot water infrastructure microbiome. And this is definitely of interest because a lot of these pathogens tend to colonize the hot water systems, but also because we invest a tremendous amount of energy in heating our water. And so in the US, we actually recommend a lower temperature setting of 48 degrees to save energy, but it's also in some healthcare settings to prevent scalding risk. Whereas other countries like Canada and Australia recommend a higher temperature to keep pathogens at bay. And so here, William Rhodes, another PhD student, led the way constructing this intricate plumbing rig that let us look at the relationship between the water heater setting as well as the frequency of water use. So you can get into water conservation scenarios and whether the pipes are up and down and how this influenced occurrence of Legionella. And this was published recently in the special issue of Microbiome that Jack Gilbert co-edited. And here you can see that we had a control rig, so there were 2 of them. The control stayed at 40 degrees Celsius, while with the experimental rig, we increased the temperature with time to see what would happen. We had triplicate pipes, and it's really fascinating that how different the water is at the tap than what's in the water heater. So you might turn up the water heater pretty hot, but by the time it gets to the tap, it spends most of its time at a pretty cool temperature. And some of the results here. So our control rig recirculating line that was at the warmer temperature, kind of a worst case scenario, is set at 40 degrees, grew a lot of Legionella. And then our experimental rig, as we elevated the temperature to 60, we did have a couple logs removal of Legionella. And then this is just showing that there weren't really much detectable Legionella in the influent, kind of around our detection limit. So it's a regrowth issue. It was something about the premise plumbing, encouraging them to grow there. And a lot of data here where we tried to track the Legionella under various conditions in this heat map. I'll just point out some trends. So one is I'm showing the control rig on the left that had the warmer, I guess cooler temperature. It was warm, this one's hot. But basically you had more growth of Legionella at that ideal temperature as you might expect. Now the water use frequency, we were curious what would happen here. And so in the control one, which was ideal for Legionella to grow, more frequent water use, instead of flushing the line and cleaning it out, was really just delivering more nutrients to the Legionella. Whereas in the hotter one, see, this was still the warm temperature. As we got hot, you were evening things out or delivering the hot water and having some disinfecting effect. And here was serendipity in action. Seems that 51 degrees Celsius and low water use is something to watch out for. There was something about that. We referred to it as an ecological sweet spot that seemed to select for Legionella. Maybe it sort of simulated the culturing method where you heat shock it, but then give it time to recover and grow. Looking a bit at the microbiome and what happened in these systems. So I guess not surprisingly, as the temperature went up, we saw a shift in the microbiome composition. And then comparing the bulk water in blue and the biofilm in red, we saw that as the temperature increased, that the composition of the biofilm and bulk water were starting to converge. So we're starting to play around with some metagenomics, and I really think this is a must as we move the field forward. So this involves extracting the DNA and directly fragmenting and sequencing it, and you can see so much more that way. Now, maybe a bit too much, because then you're seeing all the genes, including housekeeping and everything else under the sun, and it becomes a bit of an analytical nightmare. But we're working on it. We've teamed up with some computer scientists, and it's letting us zoom in on functional genes of interest, like the antibiotic resistance genes, functional genes involved in corrosion, other factors, and just a little bit of what this kind of data looks like. So again, looking at this hot water rig and a few key conditions, we saw that the hot water was distinct from the influent cold water in terms of taxonomy. So we're again looking at taxonomy like we just looked at, but from a whole different perspective. Temperature setting did matter, and the recirculating water was different from the stagnant water. And we did have shifts in the microbiome, both with temperature but also with time. And that's why it was really important to have a control rig operating in parallel to normalize to. And so we can do essentially the same analysis, but looking at the functional gene profile. And lo and behold, we see a similar trend. And you can zoom in on all sorts of functions of interest. We can just have a field day here. But one example is hydrogen metabolism. We're really interested in this because we know that water heaters, they're essentially like a microbial ecosystem. And one of the things is that when there's corrosion, they're emitting hydrogen. And that's something that can stimulate the hydrogen oxidizing microbes, which are autotrophs. And they can grow and fix carbon and just have fun in there. Implications. So we're looking at green buildings, has been talked about. And there are definitely some concerns. So water conservation is great, but that also translates into stagnation, which is not so great. And we had a chance to do a survey of some green buildings and saw several concerns, including water ages that were very long, chlorine residual that was completely absent, and elevated Legionella markers. And we're very interested in working with folks to understand what this means in terms of the human microbiome of the occupants. Another area that we're working on is recycled water. Another great idea. It's going to help with water sustainability. Essentially, you take treated wastewater, treat it to a very high standard, and you can use it for non-potable, like irrigation purposes, but we're also looking very seriously at potable reuse. And you can treat it to the highest standard, but it's still a chemically different water by nature, and it's going to behave differently in pipes. And so we have some data here showing that there's potentially more opportunity for Legionella to regrow in the recycled water. And we have data suggesting that antibiotic resistance is a concern as well. This is metagenomic data where we could look at a whole suite of antibiotic resistance genes in these different pipe systems fed, in this case, direct potable reuse water. So this is water treated to the standard of drinking, for drinking purposes. And interestingly, a lot of antibiotic resistance genes hanging out in biofilms. So what's our vision? We laid out part of this in a recent paper on probiotic control of pathogens, bit provocative, but the idea is to advance water sustainability, but also to not forget our primary mission of protecting public health, and in doing so, to embrace the microbiome, so to speak, and to not try to just futilely eradicate it, because that's going to be a losing battle. And so even bigger picture than that, as water engineers, we essentially eradicated typhoid just by chlorinating and filtering the water. Could we have a similar effect? So here's rates of antibiotic resistance increasing. Could some actions like best management practices and how we treat and distribute our water reverse trends of antibiotic resistance, or reverse trends of opportunistic pathogen infections, or help contribute to a healthy microbiome? So here's our Flint water study team with 300 kits that the students worked around the clock assembling to send out to citizens. So here's an example of citizen science and folks ready to take action. And so the question to us all here is, are we ready to take action? Thank you.
Thank you. Yeah, let's thank all 4 speakers.